Chapter 8 DNA Fingerprinting and Forensic Analysis. A biotechnician examines DNA fingerprints looking for similarities and differences between individuals. After completing this chapter, you should be able to define DNA fingerprinting, outline the process of collecting and preparing a DNA sample to be used as evidence, list some factors that can degrade DNA evidence and some of the precautions required to maintain the reliability of DNA evidence. Describe the polymerase chain reaction, PCR, method is used to produce a DNA fingerprint. Explain how short tandem repeat, STR, profiles are compared in DNA fingerprint analysis. Explain how a DNA fingerprint can be used to exclude a suspect. Examine 2 or more DNA fingerprints in the lab to determine if they match. Describe the use of DNA fingerprinting techniques in establishing familial relationships. List some of the uses of DNA analysis in biological research. Discuss some of the ethical issues surrounding DNA fingerprinting. Forensic science is the intersection of law and science. It can be used to condemn the guilty or exonerate the innocent. Many court cases hinge on scientific evidence provided by forensics. Throughout the years, scientists have developed technologies to uncover facts in criminal investigations, and the law has been quick to embrace these new technologies as they became available. For example, in the late 1800s, efforts to fight crime were given a boost by a new technology, photography. Thanks to the invention of the camera, it became possible to depict criminals in custody so accurately that images could be used as references. Photographs were certainly an improvement over verbal descriptions and hand-drawn wanted posters, but they still had severe limitations. Criminals found many ways to alter their appearance via haircuts, the addition of facial hair, or use of eyeglasses that could make identification based on a photograph almost impossible. A little more than 100 years ago, scientists discovered that the tiny arches and whorls in the skin of human fingertips could be used to establish identity. After a single bloody print on the bottom of a cash box helped solve a murder in England, the process of inking a suspect's fingers and collecting a set of prints became routine. The Federal Bureau of Investigation, FBI, the Central Intelligence Agency, CIA, and other law enforcement agencies amassed huge collections of these prints. At first, clerks were responsible for painstakingly examining the prints visually, looking for matches. With the development of the computer, however, the sorting process became less tedious and more reliable. But fingerprints are not always available, they can be wiped away, and gloves can be worn to avoid leaving them behind. In 1985, a revolutionary new technology emerged as an important forensics tool. Instead of counting on smudged fingerprints left at the scene of a crime, investigators could look at a new kind of fingerprint, the unique signature found in each person's genetic makeup. In this chapter, we look at a description of DNA fingerprints and discuss what makes each person's DNA unique. Then we learn the processes used to collect DNA samples and to produce DNA fingerprints for analysis. We compare DNA profiles and learn what it takes for a DNA match to occur. Next we consider examples that illustrate the value and vulnerabilities of DNA as evidence. We'll then look at how DNA profiles can be used to establish familial relationships, focusing on the use of mitochondrial DNA to provide proof of kinship. Finally, because DNA profiles are not limited to humans, we look at the uses of DNA analysis to establish the origin of valuable plant crops, help to enforce laws to protect wildlife, and perform biological research. Forecasting the future. The more that is known about a patient's DNA sequence, the easier it is to make predictions about what types of drugs will be effective in treating that patient's genetically based conditions. Sequencing an individual's DNA to figure out the best course of drug treatment differs from earlier tests, which serve only to diagnose certain genetic diseases, such as prostate cancer, cystic fibrosis, and diabetes. Now genetic sequencing will have to become faster and cheaper in order to develop diagnostic tests that allow for personalized therapies on a large scale. But recent advances are producing second and third generation innovations that may reduce the cost of human DNA sequencing by 2/3 in as little as 10 years. Medicare and private insurers have been slow to accept the cost of reimbursing patients for DNA sequencing, but strong demand for personalized medicine in pharmaceutical research could increase acceptance of these new costs. In fact, it is likely that in the near future, the U.S. Food and Drug Administration, FDA, will require companion diagnostics, in the form of DNA sequencing of phase trial participants, for every drug being tested to treat genetic disorders, see Chapter 12. Additionally, in June 2010, the FDA put 5 genetic test makers on notice that they must get federal approval before marketing their products directly to consumers. The biology of how DNA variations actually cause certain diseases is still being actively researched, but these efforts will undoubtedly lead to the need for more DNA testing on an individual level. Making genetic tests the standard of care has the potential to significantly reduce medical costs and increase safety. 8.1 What is a DNA fingerprint? We know that every human being carries a unique set of genes. The chemical structure of DNA is always the same, but the order of the base pairs in chromosomes differs in individuals. The novel assemblage of the 3 billion nucleotides formed into 23 pairs of chromosomes gives each of us a unique genetic identity. We also know that every cell contains a copy of the DNA that defines the organism as a whole, even though individual cells have different functions. Cardiac muscle cells keep our hearts beating, neurons transmit the signals that are our thoughts, etc. These two aspects of DNA, its uniform nature in a single individual and the genetic variability between individuals, make DNA fingerprinting possible. Because every cell in a body shares the same DNA, cells collected by swabbing the inside of a person's cheek will be a perfect match for those found in white blood cells, skin cells, or any other tissue. Figure 8.1 Restriction fragment length polymorphisms, RFLPs, can be used to produce DNA fingerprints. Restriction enzymes recognize a short sequence of nucleotides and cut the DNA. Random mutations interfere with enzyme recognition, creating different lengths of DNA, not cut if mutated. The pattern of these cut fragments, when separated on a gel by electrophoresis, can be used to determine similarities and differences. Human DNA fingerprinting has moved to STR analysis largely because of the need for considerably less DNA for the PCR process. Individual 1 in the figure has 3 cut sites in his DNA, while individual 2 has only 2. The bending patterns resulting from the electrophoretic separation shows the differences. How is DNA typing performed? Only 0.1% of DNA, about 3 million bases, differs from person to person. These variable regions can generate a DNA profile of an individual, using samples from blood, bone, hair, or other body tissues containing a nucleus with DNA. In criminal cases, this generally involves obtaining samples from crime scene evidence in a suspect, extracting the DNA, and analyzing it for the presence of a set of specific DNA regions. There are 2 main types of forensic DNA testing, one based on restriction fragment length polymorphism, RFLP, and the other on the polymerase chain reaction, PCR. See Figure 8.1 for an illustration of an RFLP. Generally, RFLP testing requires larger samples than does PCR, and the DNA must be intact and undegraded. The FBI accepts RFLPs of 4 types for case comparison. Crime scene evidence that is old or present only in small amounts is often unsuitable for RFLP testing. Warm, moist conditions may accelerate DNA degradation, rendering it unsuitable for RFLP testing in a relatively short period of time. Because of this, RFLP is not used as often for DNA testing as it once was. PCR-based testing requires less DNA than RFLP testing and is still effective if the sample is partially degraded. However, PCR-based tests are also extremely sensitive to contamination by foreign DNA at the crime scene and within the laboratory. Fortunately, it is not necessary to catalog every base pair in an individual's DNA to arrive at a unique. Figure 8.2 Anatomy of a DNA microsatellite. A microsatellite is a variable number of repeated nucleotides, like TTA, that occur in specific locations in the genome. Using primers for the flanking regions on the ends of the microsatellite allows for amplification using PCR, from both directions. Individuals inherit a specific number of these repeats from their parents, but the number of repeats varies for unrelated individuals, forming a distinct pattern or DNA fingerprint. Fingerprint. Instead, DNA profiling depends on only a small portion of the genome. Every strand of DNA is composed of both active genetic information, which codes proteins, and inactive DNA, which does not. Some of these inactive portions contain repeated sequences of between 1 and 100 base pairs. See Chapter 3 for a review. These sequences, called variable number tandem repeats, VNTRs, are of particular interest in determining genetic identity. Every person has some VNTRs that were inherited from his or her mother and father. No person has VNTRs that are identical to those of either parent. This could only occur as a result of cloning. Instead, the individual's VNTRs are a combination of repeats of those of the parent's DNA regions in tandem. The uniqueness of an individual's VNTRs provides the scientific marker of identity known as a DNA fingerprint. DNA fingerprints produced by PCR are usually restricted to detecting the presence of microsatellites, figure 8.2, which are 1 to 6 nucleotide repeats dispersed throughout chromosomes. Because these repeated regions can occur in many locations within the DNA, the probes used to identify them complement the DNA regions that surround the specific microsatellite being analyzed. The small size of these repeated segments has resulted in the term short tandem repeat, or STR. The FBI has chosen 13 unique STRs to test for DNA profiles contained within their combined DNA index system, or CODIS, as seen in Figure 8.3. Figure 8.3 Human Short Tandem Repeat Patterns. The FBI has chosen 13 unique street regions, shown on human chromosomes, for analysis and comparison in its library of DNA fingerprints. DNA primers for the flanking regions of these sites are commonly found in PCR kits that amplify these sites. After DNA sequencing, the results indicate the number of repeats for each homologous chromosome at that site and constitute a DNA fingerprint for that individual. 8.2 Preparing a DNA fingerprint. Preparing a DNA fingerprint requires specimen collection, DNA isolation and quantification, and PCR amplification. We discuss these steps in detail on the following pages. Specimen collection. Crime scene investigators routinely search for sources of DNA, dirty laundry, a licked envelope, a cigarette butt, or anything else that might be a source for human cells left behind. Tiny blood stains or a trace of saliva is often all it takes to crack a case. Every living thing has DNA, so every crime scene is full of sources of possible contamination. For this reason, scrupulous attention to detail is required in collecting and preserving evidence. To protect the evidence, workers at a crime scene must take the following precautions. See FBI website for a full list of precautions. Wear disposable gloves and change them frequently. Use disposable instruments, such as tweezers and swabs. Avoid talking, sneezing, and coughing to prevent contamination with microdroplets of saliva. Avoid touching anything that might contain DNA, like their own body parts, while handling evidence. Air dry evidence thoroughly before packaging. Microbes can contaminate a sample. Store evidence in specifically designed materials. Plastic bags can retain damaging moisture. As this partial list shows, the enemies of evidence are everywhere. Sunlight and high temperatures can degrade DNA. Bacteria, busy doing their natural work as decomposers, can contaminate a sample before or during collection. A small amount of moisture can damage and ruin a sample. DNA fingerprinting is a comparative process. DNA from the crime scene must be compared with known DNA samples from the suspect. The ideal specimen used to compare evidence is 1 ml or more of fresh whole blood treated with an anti-clotting agent called ethylene diamine tetrahydric acid, EDTA. This quantity of whole blood contains an adequate amount of leukocytes with nuclear DNA for testing. Unlike erythrocytes, which have no nuclear DNA, leukocytes contain the normal complement of human chromosomes. If a clean specimen cannot be obtained from the known source, all is not necessarily lost. DNA has been retrieved and successfully analyzed from samples that were a decade old by amplifying the DNA used for comparison by PCR. Extracting DNA for analysis. After a sample is collected from the known source, a lab technician is responsible for determining its genetic profile. First, the technician extracts the DNA from the sample. DNA can be purified either chemically, using detergents that wash away the unwanted cellular material, or mechanically, using pressure to force the DNA out of the cell. Once the DNA is extracted, the technician must follow several steps to transform the unique signature of that DNA into visible evidence. PCR and DNA amplification. Several thousand cells are required to do an RFLP analysis, more than can often be collected at a crime scene. Imagine a case in which the only evidence is the saliva on an envelope flap or a blood stain invisible to the naked eye. Because of this PCR is now normally used to grow or amplify the sample into an amount that can be analyzed. PCR is much like a photocopier for DNA. The first step involves locating the portions of DNA that can be useful for comparison. Primers, or short pieces of DNA, are the tools used to find those portions on each DNA strand in the 5 to 3 direction, see arrows in figure 8.4. The primers behave much like probes and seek out complementary sections of the DNA. Once the complementary segment of DNA is located, copying begins using a device called a thermal cycle, see Chapter 3. Figure 8.4 illustrates PCR as used in DNA amplification for fingerprinting. With the aid of the enzyme Taq polymerase and DNA nucleotides, cycles of heat and cold cause the DNA to separate and replicate repeatedly. Within hours, the DNA segments can be increased by millions or even billions. That rapid growth presents a possible problem. PCR is very sensitive to contamination, and a small error in field or laboratory procedure can result in the duplication of a useless DNA sample a million times. STR analysis. After STRs are amplified by PCR, the alleles are separated and detected using capillary electrophoresis, which separates the lengths of amplified DNA, allowing the number of repeats in each of the 2 alleles on homologous chromosomes to be determined. An STR contains repeated numbers of a short, typically 3 to 4 nucleotide, DNA sequence. The number of repeats within an STR is referred to as an allele. For instance, the STR known as D7S820, found on chromosome 7, contains between 5 and 16 repeats of GATA. Therefore, there are 12 different alleles possible for the D7S820 STR sequence. An individual with D7S820 alleles 10 and 15, for example, would have inherited a copy of D7S820 with 10 GATA repeats from one parent and a copy of D7S820 with 15 GATA. Figure 8.4 Amplifying DNA at specific sites by PCR. DNA primers for the flanking regions of Cody's sites results in DNA amplification at specific STR sites. Many PCR kits contain reaction beads with multiple primers, making it possible to amplify DNA for multiple sites simultaneously. Repeats from the other parent. There are 12 different alleles for this STR, creating 78 different possible genotypes, or pairs of alleles, for this one site. The large number of variations for each STR provides the variability that is needed to distinguish different individuals using DNA fingerprints. Figure 8.5 shows alleles of all 13 STR sites accepted by the FBI for Cody's. 8.3 Putting DNA to use. DNA fingerprinting is similar to old-fashioned fingerprint analysis in an important way. In both cases, evidence collected from a crime scene is compared with evidence collected from a known source. During evaluation of the samples, analysts are looking for alignment of the bands. If the samples are from the same person, or from identical twins, the bands or dots should line up exactly. All tests are based on exclusion. In other words, testing continues only until a difference is found. If no difference is found after a statistically acceptable amount of testing, the probability of a match is high. See if you agree with the exclusion of one individual as shown in Figure 8.6. Figure 8.5 Gene scan display of all 13 STRs accepted by CODIS. Utilizing capillary electrophoresis to detect the amplified STR microsatellite DNA sequences, it is possible to determine the number of STRs. By comparing the patterns obtained as evidence, it can be determined if the DNA patterns are exactly the same or different. Note that none of the DNA fingerprints shown are exact matches. As we will see, such clear-cut visual evidence can be invaluable to crime investigators. Next we consider a few examples of DNA profiling in action. The Narborough Village murder case led to a new method of DNA separation. The first reported use of genetic fingerprinting in a criminal case involved the sexual assault and murder of a schoolgirl in the United Kingdom in 1983. Sir Alex Jeffreys and his colleagues, Dr. Peter Gill and Dr. Dave Werrett, developed techniques for extracting DNA and preparing profiles using old samples of human tissue. Gill also developed a method for separating sperm from vaginal cells, which allows fingerprints to be run on vaginal cells first and then sperm cells in order to provide comparison samples, Figure 8.7. In this method, a detergent breaks open vaginal cells but leaves sperm cells intact. Without these developments, it would be difficult to use DNA evidence to solve rape cases because sperm cells could not be located to compare them against known samples. In this case, Jeffreys and his colleagues compared DNA evidence collected from the murder scene with a semen sample from an earlier similar rape and murder, and the analysis indicated that both crimes were. Figure 8.6 Who is excluded by DNA comparison? The DNA profiles shown below were taken from a crime scene. Knife stains A and B were found on the blade of a knife used in the crime, while stain C was found on the handle of the knife. All three stains were amplified for DNA analysis. 2 individuals were tested for a match to the DNA profiles. Read the conclusion from the figure and see if you agree with the analysis. Case mock-up, results. Conclusions. Dorothy Smith can be excluded from contributing to the DNA found on the knife blade stains A and B. Jane Doe cannot be excluded from contributing to the DNA found on the knife blade stains A and B. Sample ID D3S1358 VWA FGA ML D8S1179 D21S11 D18S51 D5S818 D13S317 D7S820 Jane Doe, reference sample 15, 16 16, 19 18, 22 X 11, 12 30, 31.2 14 9, 12 9, 12 8, 11 Dorothy Smith, reference sample 15, 18 16, 17 20, 21 X 10, 13 31, 31.2 12, 16 10, 12. 11, 12. 8. Knife blade stain A. 15, 16. 16, 19. 18, 22. X. 11, 12. 30, 31.2. 14. 9, 12. 9, 12. 8, 11. Knife blade stain B. 15, 16. 16, 19. 18, 22. X. 11, 12. 30, 31.2. 14. 9, 12. 9, 12. 8, 11. Figure 8.7 Isolation of sperm DNA or vaginal cell DNA from mixed sources. DNA from a sperm cell can be fingerprinted to be compared with the profile gotten from a sperm sample obtained on a vaginal swab. The buffer, containing dithiothiopol, DTT, will break the sperm cell membrane and can be used to detect sperm DNA after vaginal DNA has been analyzed separately in mixed evidence, owing to the ability of the sperm cell membrane to resist disruption until the addition of DTT. Committed by the same man. At this point, the police had a prime suspect. However, when the DNA evidence was compared with a DNA sample from the suspect's blood, the DNA did not match. The prime suspect was therefore excluded from consideration. The investigation continued. The police conducted the world's first mass screening of DNA, collecting 5,500 samples from the district's male population. Using simple blood typing tests, all but 10% of the initial group were quickly excluded, using RFLP analysis methods. After many grueling hours of analysis, the investigation had come to a standstill. None of the remaining profiles matched that of the rapist/killer. Then came the lucky break. A man was overheard saying that he had given a sample in the name of a friend. When the man who had evaded the mass testing was apprehended, his DNA was analyzed and found to be an exact match for the DNA in the semen specimens from the crimes. The suspect confessed and was sentenced to life in prison. This case highlights one of the important limitations to the use of DNA evidence. Unless there is a known sample to be used as a comparison, identity cannot be established. In the example shown in Figure 8.8, the blood samples from the victim and the suspect are known. By comparing these two known DNA fingerprints, investigators were able to place the suspect at the scene of the crime based solely on the DNA fingerprints of the blood found on the suspect's clothes. Is eyewitness testimony more reliable than DNA testing? DNA evidence was first used in the United States in 1987 and has since helped to resolve thousands of cases. A particularly important use of DNA evidence is to refute other erroneous evidence that has sometimes led to false convictions. DNA evidence is especially valuable when it is used to expose faulty eyewitness testimony. Eyewitness testimony, which might seem to be the gold standard of evidence, is actually quite fallible. Figure 8.8 Example of an exclusion based on one STR from a suspect. At locus D5S818, chromosome 5, person A has 9 repeats from his mother and 12 repeats from his father. Person B has 8 repeats from both his parents. Sample ID D5S818. Person A 9, 12. Person B 8, or 8,8. You decide, should DNA fingerprinting be sped up to apprehend repeat offenders? Some criminals are arrested, spend less than a day in jail, and then commit crimes while they are out on bail. If police could quickly test suspects' DNA, to see if it matches entries in crime databases, they might be able to keep dangerous criminals in jail. Currently, most genetic tests take 24 to 72 hours, and by the time that the results are back, the suspects may have been released. A new test could make it almost as easy as matching fingerprints to compare DNA from people arrested for crimes with other DNA samples from crime scenes stored in forensic databases. With this test, police could check on whether a person's DNA matched that found at past crime scenes while suspects were still being processed. A report on the FAST forensic test appears in the August 5, 2010, issue of the journal Analytical Chemistry. To increase the speed of forensic DNA testing, the scientists built a chip that can copy and analyze DNA samples taken from a cotton swab. Forensic technicians collect DNA by swabbing a suspect's mouth, mixing the sample with a few chemicals, and warming it up. The DNA testing lab on a chip does the rest. The entire process takes only 4 hours at present. Is testing a suspect's DNA upon arrest for speedy results a reasonable way to catch repeat offenders, or is it an invasion of privacy? You decide. For example, in 1988, Victor Lopez, the so-called Forest Hills rapist, was tried for the sexual assault of 3 women. All 3 women had described their assailant as a black man when they reported the crime to the police. Because Lopez was not black, concerns were raised that this was a case of mistaken identity. Was Lopez an innocent man falsely accused by the system? The blood of the accused was analyzed, it was then compared with sperm left at the scene and the DNA was a match. Lopez was found guilty of the attacks despite the contradictory eyewitness testimony. Now let's look at how much of what we have learned is admissible as evidence. World events lead to the development of new technologies. When major disasters occur and large numbers of people need to be identified, DNA comparisons are not enough. New methods of cataloging evidence have resulted in faster and more efficient comparisons to distinguish individuals. World Trade Center. Shortly after the Twin Towers of the World Trade Center in New York City were destroyed by the terror attacks of September 11, 2001, forensic scientists came together and rapidly accelerated efforts to use DNA-based techniques to identify the remains of victims. They were confronted with a tremendous amount of debris at the site, dangerous working conditions, heat and microbial decomposition of remains, and hundreds of thousands of tissue samples from the nearly 3,000 individuals lost. These issues made it evident that new strategies would need to be employed to quickly prepare and organize DNA profiles and compare them with DNA profiles from relatives. How would scientists establish DNA identity for those who perished in over 1.5 million tons of rubble? State agencies such as the New York Department of Health, and the Medical Examiner's Lab, and several federal agencies including the U.S. Department of Defense, National Institutes of Health, and the U.S. Department of Justice immediately responded to help with this task. Within 24 hours after the disaster, the New York City Police Department had established collection points throughout the city where family members could file missing persons reports and provide cheek cell swabs for DNA isolation. Personal items, combs, toothbrushes, from the missing were also collected for DNA profiling. Myriad Genetics, Inc., Gene Codes Forensics, DNA View, Celera Genomics, Bode Technology Group, and Orchid Gene Screen were among the companies assisting in this effort. Several other companies were involved in developing new software programs to help match samples submitted by family members to DNA profiles obtained from World Trade Center victims. Available tissue samples primarily consisted of small bone fragments and teeth, which provided fragmented DNA owing to degradation by the intense heat at the site. Because of this, forensic scientists primarily used STR, mitochondrial DNA, mtDNA, and SNP analysis of DNA fragments to develop profiles. DNA analysis was conducted on over 15,000 tissue samples, although less than 1,700 of the estimated 2,819 people who died at the site were ultimately identified. This tragedy forced the development of new forensic strategies for analyzing and organizing remains recovered from the site and, most importantly, provided closure for a number of families that lost loved ones in the attack. South Asian Tsunami. The South Asian tsunami of December 2004 was a tragedy that claimed over 225,000 lives and devastated areas of Indonesia, Sri Lanka, and Thailand. You decide crime-fighting tool or invasion of privacy. In the Narborough murders, many individuals were tested because they all fit a general description. They were young men who lived in the area of the murders. This strategy led to the conviction of the killer, but mass testing, especially on the basis of a general description, is extremely controversial. Opponents point out that more than 5,000 innocent men were called on to give a sample of their blood during the investigation, and the guilty individual very nearly escaped detection despite these efforts. Such mass testing without due cause is now prohibited both in the United States and the United Kingdom. However, many people still believe that even the routine quest for DNA evidence during a criminal investigation continues to undermine the fundamental right to privacy. Of particular concern are DNA profiling databases. Computer searchable collections of DNA data are now authorized by all 50 states. Many states have also registered the DNA profiles they collect on CODIS, the combined DNA index system run by the FBI. In addition to profiles of convicted offenders, CODIS contains a compilation of unidentified DNA profiles taken from crime scenes, and some advocates are calling for adding DNA profiles of all people arrested for any offense. Some states have even proposed that DNA profiles be taken from all individuals at the time of birth to create databases that might be used to identify criminals by matching DNA profiles from their relatives. In early 2007, the federal government proposed that DNA profiles from more than 400,000 illegal immigrants and detainee captives in the war on terrorism be added to CODIS. By comparing the databases, law enforcement officers can identify possible suspects when no prior suspect exists. Visit the CODIS FBI website to see how the information is used in solving crimes. Opponents do not argue with the potential usefulness of the databases, but they are concerned that the technology could be abused. They point out that taking a blood sample is a more invasive process than taking a set of fingerprints. In some cases, the courts have agreed and determined that DNA collection is a violation of state and federal laws prohibiting unreasonable search and seizure. There is also the possibility that DNA information could someday be used to discriminate against job seekers or those applying for health insurance. Even though profiles are based on junk DNA and provide no information about genetic diseases or other traits, the original sample does contain an individual's complete DNA. On this basis, some demand that all samples be destroyed after investigation of a specific case is completed. Proponents of DNA collection point out that the databases are regulated and secure, and samples are not obviously identified with the name of the source. Only trained professionals collect the blood samples, and the procedure presents no significant health risk to the donor. Is the routine collection of blood and the compilation of DNA databases a reasonable tool in the effort to fight crime or an unwarranted invasion of privacy? Should investigators be allowed to conduct the type of mass screening used in the Narborough case? You decide. Michigan-based company Gene Codes modified their software system, called Mass Fatality Identification System, (MFISIS), to help with the Thailand tsunami victim identification effort. Because MFISIS was essentially built in response to the 9/11 tragedy, Gene Codes did not have to write entirely new software and were able to customize MFISIS as necessary. In addition to analyzing mitochondrial DNA, see Section 8.6, MFISIS incorporated male-specific variations in the Y chromosome, called YSTRs, to aid in the identification of individuals. Within 3 months, approximately 800 victims had been identified. Gene Codes also established the DNA Show project. Show is the Hebrew name for the Holocaust, an effort to use MFISIS to establish a genetic database of Nazi-era Holocaust survivors with an overall goal to try and reunite an estimated 10,000 post-war orphans around the world. 8.4 DNA and the Rules of Evidence. Before DNA fingerprints could ever be used in a court of law, DNA fingerprinting had to meet the overarching legal standards regarding the admissibility of evidence. Courts use 5 different standards to determine whether scientific evidence should be allowed in a case. The test used depends on the jurisdiction. When a new technique or method is used to collect, process, or analyze evidence, it must meet one or several of these standards before the evidence is accepted. The relevancy test, Federal Rules of Evidence 401, 402, and 403, essentially allows anything that is deemed to be relevant by the courts. The Frye Standard, 1923, requires that the underlying theory and techniques used in gathering evidence have been sufficiently used and tested within the scientific community and have gained general acceptance. This is known as a general acceptance test. The Coppolino Standard, 1968, allows new or controversial science to be used if an adequate foundation can be laid, even if the profession as a whole is not familiar with the new method. The Marx Standard, 1975, is basically a common-sense test that requires that the court be able to understand and evaluate the scientific evidence presented. This is sometimes referred to as the no-jargon rule. The Daubert Standard, 1993, requires special pretrial hearings for scientific evidence. Under the Daubert Standard, any scientific process used to gather or analyze evidence must have been described in a peer-reviewed journal. The goal is to make certain that the scientific methods and expertise used to provide evidence are trustworthy. DNA fingerprinting and the chain of evidence. DNA analysis was a relatively new forensic tool when the Los Angeles Police Department used it in the infamous O.J. Simpson trial in 1994. Simpson's ex-wife, Nicole Brown Simpson, and her friend Ronald Goldman were murdered, and Simpson was the primary suspect. 45 samples were collected for DNA analysis from the crime scene. During pretrial proceedings, it was announced that the DNA collected at the crime scene matched that of O.J. Simpson. Defense lawyers immediately attacked the procedures used in collecting, labeling, and testing the evidence. During the trial, the defense showed a videotape of the sample collection methods and drew on expert testimony to establish doubt about the reliability of the evidence submitted. The defense stressed that contamination could have occurred when a technician touched the ground, when plastic bags were used to store wet swabs, and when sample collection tweezers were rinsed with water between touching samples. While on the stand, one prosecution witness admitted to mislabeling a sample. The possibility that the evidence might be tainted was obvious to both the court and the jury. As a result, the DNA evidence, which had been expected to make the case for the prosecution, was not effective. O.J. Simpson was found not guilty. When the chain of evidence is broken, when the rules of evidence are not followed, DNA samples lose their value in court. Human error and sources of contamination. One of the greatest threats to DNA evidence is human error. Earlier, we reviewed some of the precautions that crime scene investigators take to preserve and collect DNA evidence. A sneeze, improper storage, failure to label every single sample. These seemingly small mistakes can result in the destruction of evidence. Even if DNA evidence is not degraded by careless handling or bad conditions, it can be disregarded if the chain of evidence is suspect. As we saw in the example of the O.J. Simpson trial, the chain of evidence requires that the collection of evidence be systematically recorded and access to the evidence must be controlled. Crime scene sample collection presents challenges, but even the collection of samples in a controlled environment, such as a morgue, is also problematic. Studies of morgue tables and instruments have found that the DNA of at least 3 individuals is often present. That DNA could certainly confuse the results of any analysis undertaken on samples collected in that environment. DNA evidence is also vulnerable to damage during analysis. For example, DNA from the technician's body or from other sources can be inadvertently added to the sample. Defined standards of laboratory practice and procedure can help guard against these errors. However, when DNA evidence was a new idea, laboratories were not regulated and serious errors were made. Consider the case of New York v. Castro, which involved the murder of a woman and her 2-year-old child. The prosecution presented autorads, RFLPs, produced with the Southern blot method of the suspect's DNA and crime scene DNA and claimed that they matched. However, as an expert witness for the defense pointed out, the bands most certainly did not match. In this case, technicians made errors during their analysis and evaluation of the autorads. Fortunately, because the standards for the admissibility of evidence are high and the defense had access to DNA experts as well, these errors did not lead to a fatal miscarriage of justice. The suspect could not be linked to the crime with this evidence. One step to ensure the reliability of DNA is to make certain that laboratories that process samples adhere to consistently high standards. The American Society of Crime Laboratories Directors, USCLD, the National Forensic Science Technology Center, NSFTC, and the College of American Pathologists, CAP, all provide accreditation to forensic laboratories. Proficiency testing of technicians has become a basic requirement for employment. These exams include blind tests in which the technician is unaware that the sample being processed is actually a test sample submitted by the certifying organization. In addition, the FBI has developed a standard operating procedure for the handling and DNA typing of evidence in criminal cases. With clear. Figure 8.9 Paternity Dispute. A man has claimed not to be the father of a child, but DNA allelic comparison shows that the child received one allele from each parent. Follow the arrows from the parents to the child to see how each parent contributed one of the two alleles, bands, that show in the DNA fingerprint of the child. Guidelines in place and thorough training of those responsible for collecting and preserving evidence. The reliability of DNA evidence presented in court proceedings should increase. DNA and juries. The use of DNA evidence also presents another challenge. To be useful, the science must make sense to the jury evaluating the case. Because DNA evidence is statistical in nature, the results can be confusing to a person without sufficient education, especially when large numbers are involved. When members of a jury panel hear there is 1 chance in 50 billion that a random match might occur, it is possible they may focus on that one possibility and discount the overwhelming odds against its happening. Oddly enough, the same jurors might accept a conventional fingerprint as more reliable evidence, even though studies have concluded that such evidence is actually less statistically valid. Making DNA evidence clear and comprehensible to jurors is not an easy task. And if the DNA evidence is not clearly understood, it may be disregarded. 8.5 Familial relationships and DNA profiles. Crime solving is not the only forensic application of DNA fingerprinting. Because family members have a small amount of DNA in common, relationships can be conclusively determined by comparing samples between two individuals. An obvious use of this is in paternity testing, figure 8.9. Every year, roughly 400,000 paternity suits are filed in the United States, Canada, and the United Kingdom. Given samples from the child and adults involved, verifying the child's parentage, and resolving child support or custody disputes is relatively easy. However, reproductive technologies including in vitro fertilization, artificial insemination, and surrogacy have introduced a few quirks into custody and paternity. DNA testing has helped untangle cases in the courts that involved sperm mix-ups in fertility clinics, for example. Thanks to amniocentesis, as shown in figure 8.10, it is possible to verify a child's parentage even before birth. Mitochondrial DNA analysis. Mitochondrial DNA analysis can be used to examine DNA from samples that cannot be analyzed by RFLP or STR. Nuclear DNA must be extracted from samples for use in RFLP, PCR, and STR, but mtDNA analysis uses DNA extracted from a cellular organelle called a mitochondrion. Older biological samples that lack nucleated cellular material, such as hair, bones, and teeth, cannot be analyzed with STR and RFLP, but they can be analyzed with mtDNA. All daughters have the same mitochondrial DNA as their mothers because the mitochondria of embryos come from the mother's egg cell. The father's sperm contributes only nuclear DNA and no mitochondria. Comparing the mtDNA profile of unidentified remains with the profile of a potential maternal relative can determine whether they share the same mtDNA profile and are related. Figure 8.10 DNA for paternity testing. For disputed paternity suits, it is possible to draw a few fetal cells from the fluid that surrounds the fetus, amniotic fluid, without harming the growing fetus. When cultured, these cells can be a source for DNA extraction and fingerprinting. When compared with the DNA of the suspected father, exclusion or inclusion of the individual can be determined. mtDNA remains virtually the same from generation to generation, changing only about 2% to 4% every million years due to random mutation. Consequently, relationships can be traced through the unbroken maternal line, as shown in Figure 8.11. mtDNA evidence was essential in reuniting families torn apart in Argentina during the Falklands War in the early 1980s. During this repressive regime, the military government routinely arrested and questioned anyone suspected of subversive activity. Among those arrested were a number of young pregnant women. Roughly 15,000 of those arrested and questioned simply disappeared. After the collapse of the government in 1983, the mothers and grandmothers of these disappeared women became aware that the horrible crime might well be ongoing. Babies born to their daughters while in prison had been taken and were being raised by others, often by the families of those who had murdered their parents. The mothers sought help from the American Association for the Advancement of Science, AAAS, asking if genetic testing could be used to prove the identity of the stolen children. Through the perseverance of these women and the scientists who volunteered their help, the AAAS was able to prove that at least 51 children had been illegally adopted. DNA evidence was used in court to restore the children to their living relatives. Y chromosome analysis. The Y chromosome is passed directly from father to son, making the analysis of genetic markers on the Y chromosome. Figure 8.11 Maternal DNA pattern of inheritance. Key genes for mitochondrial function, cell respiration, are located in a small DNA ring in human mitochondria. Because mitochondria are contributed by the egg, only, before fertilization, DNA can be traced through the maternal line with fingerprinting of the mitochondrial DNA. Some genes in mitochondria show variations due to mutations. See top of gel at 8.6, 4.7 versus 4.5, 4.1 kilobases, others do not. Useful for tracing relationships among males or for analyzing biological evidence involving multiple male contributors. The Y chromosome can be analyzed with nuclear DNA, usually by PCR methods. 8.6 Non-human DNA analysis. Not every legal case is a matter of human identity. Many legal questions as well as scientific quandaries have been answered by examining the DNA fingerprints of plants and animals. Expensive plants, animals, and their biological products can be protected using their DNA profiles. Identifying plants through DNA. Ginseng is a valuable herbal product. The market for it is estimated to be $3 million in the United States alone, and the demand is also high in other countries. Currently, two major herbal products are referred to as ginseng. One is native to North America and the other to Asia. Oddly, Asian ginseng is the type most often sold in American health food stores. Most American ginseng, the rarer and more valuable of the two varieties, is exported to Asia. The two types of ginseng look almost identical, but they have very different reputations. Asian ginseng purportedly boosts energy, whereas American ginseng is prized for its supposed ability to calm nerves. Manufacturers are now using DNA sequencing to help make the distinction between the two varieties. Confirming the origin of the product helps ensure quality control and protects the market for American ginseng products. This same type of analysis is performed for other plants as well. A DNA profile used in quality control of squash seeds is shown in Figure 8.12. This quality control process allows the seed supplier to guarantee. Tools of the trade. DNA profiles are advancing biological research. In addition to their use in legal proceedings, DNA profiles from animals have been used in direct biological research. In 2002, it was reported that DNA analysis of the remaining bison herds, both public and private, indicated that the majority of bison have some domestic livestock as ancestors. There is no outward sign of the hybrid nature of these animals, but their heritage is clearly evident in their DNA. As a result of this research, steps can be taken to preserve the remaining pure herds. Scientists have also found that chickadees, barn swallows, indigo buntings, and other species are not monogamous by examining the DNA of parent birds and their offspring to determine if the babies in the nest actually had different fathers. In addition, mitochondrial DNA, mtDNA, analysis has proven to be a rich source of new information about evolutionary biology. Mitochondria are estimated to mutate at the rate of 2% to 4% per million years, which allows scientists to trace changes in gene frequency through time. Using frequency changes in mtDNA, scientists are able to differentiate between similar animal species and to examine the role of base substitution during evolution. Only a nanogram of mtDNA is required for direct sequencing identification. In fact, the Eve hypothesis, which used mtDNA evidence to trace a majority of people now living on Earth to a single female ancestor from ancient Africa, is based on mtDNA analysis. The results of that study have now been compared with a similar human origins study following sequence variations in the Y chromosome. Figure 8.12 DNA profile of squash hybrids used as a quality control analysis. The anode is in the center of this gel with cathodes at the top and bottom. When the DNA from 96 seeds is loaded at the center of the gel, it will migrate to the cathodes during electrophoresis. The male parent plant donated the upper DNA band and the female donated the lower of the two bands of the hybrid offspring plant being tested. The hybrid plants all have both bands. One exception gives 98.9% quality assurance of purity in 96 seeds tested. This level of hybridity, quality control, will increase the value of the seed to a grower depending on a high level of hybridity for the growing requirements. A specific degree of genetic hybridity, thus increasing the seed's value to growers, who depend on these traits to sell their high-value crops. Another interesting paternity case of the plant world involves the ancestry of Cabernet Sauvignon grapes, which are prized for wine production. Hybrid grapes are considered inferior by some wine purists and legally excluded from bearing the prestigious distinction Appellation d'origine contrôlée in France. When scientists examined the DNA of Cabernet Sauvignon plants, it was evident that two other varieties of grapes, Cabernet Franc and Sauvignon Blanc, were the ancestors of Cabernet Sauvignon. This finding challenges the old notion that varieties derived from crossbreeding are inherently less valuable as wine grapes. Animal DNA analysis. DNA evidence has also been used to generate genetic profiles of animals. For example, in Pennsylvania, DNA fingerprinting was used to prove that a hunter had killed a bear illegally. The bear hunting season in that state is designed to protect pregnant sows. A law making it illegal to kill bears in their dens ensures that a large portion of the breeding female population will be protected from loss during the season. In this case, a witness reported seeing a hunter discharge his rifle into a bear's den. The hunter had registered his kill at the appropriate check station, without disclosing the circumstances under which it was shot, where one of the harvested bear's premolars was removed, as specified by state hunting regulations, to verify the sex and age of the kill. As part of their investigation of this eyewitness report, authorities collected blood samples from the den and compared the DNA from the den with that from the check station. The tests revealed that the DNA samples were from the same animal, even though the hunter, in an effort to evade prosecution, had claimed to have killed the bear 5 miles from the den. Because of the DNA evidence, he was found guilty. DNA profiling is regularly conducted by wildlife management authorities in such cases. Figure 8.13, for example, shows DNA profiles for 4 deer that were killed by a hunter with a permit to kill only 1 deer. Armed with the tools of DNA fingerprinting, wildlife managers have improved their ability to prove that game has been taken beyond the limits set by regulations. DNA tagging to fight fraud. A highly effective barrier to product counterfeiting and piracy is possible by using DNA as an authentication label hidden in a wide variety of products. Virtually anything of value, from fine art to sports memorabilia, can have DNA incorporated into it. The DNA labels can then be detected using PCR or hybridization techniques to verify authenticity. For example, footballs in the 2011 Super Bowl were encoded with DNA to authenticate them as official products of the National Football League. During the 2010 Sydney Summer Olympics, DNA Technologies of Halifax, Nova Scotia, tagged 34 million labels with Figure 8.13 Validation of microsatellite evidence in a deer poaching case. 2 deer, illegally shot, were found by the side of the road. A suspect had a blood stain, deer, in the trunk of his car. The DNA matched one of the deer, and the suspect was convicted, fined $2,000, and spent a year in jail. The microsatellite STRs of California mule deer had to be discovered to perform the analysis shown below. Specific DNA strands and then the labels were attached to Olympic licensed merchandise. Random inspections of merchandise vendors in Sydney revealed that nearly 15% of the merchandise sold as officially licensed was in fact counterfeit, contributing to nearly $1 million in lost sales. Earlier, you learned why DNA is a good forensic marker. These same reasons make DNA a good physical marker that is difficult to counterfeit. Typically, DNA for labels incorporate random single-stranded sequences from 20 to several thousand nucleotides long. These strands are then mixed into ink, cloth, threads, or other materials used in the product to be tagged. To determine whether an item is authentic, the portion thought to contain the DNA tag is removed and analyzed by PCR, or probes are added to the component that will fluoresce if they hybridize with the single-stranded tags in the product. DNA tagging has become a commonly used tool to combat counterfeiting. Career Profile Laboratory Technician. Laboratory technicians are commonly employed in DNA fingerprinting labs to perform tests on evidence collected from crime scenes. These technicians must be able to follow directions with great accuracy. They must take great care to keep the work area clean so as to prevent contamination. In fact, all technicians working in law enforcement labs must pass a quantitative analysis course as a condition of employment. Technicians frequently may be required to work in a special cleanroom environment wearing sanitary gowns. Salaries range from $21,000 to $35,000 for an entry-level worker to $25,000 to $60,000 for more experienced technicians. Most employers look for individuals with a bachelor's degree in biology, biochemistry, or molecular biology. Some hire applicants with a specialized associate's degree in biotechnology and laboratory experience. Hands-on laboratory experience is a critical skill in this position and may be gained through coursework, internships, and on-the-job training. However, technicians also need good math and communication skills. Writing is especially important because lab notebooks are considered legal documents. Strong computer skills are also necessary. Opportunities for advancement to research associate, especially for those with a bachelor's degree, are good. Making a difference. As the cost of sequencing the human genome becomes more affordable, the information gained from comparing multiple human genomes becomes more valuable. The Human Reference Genome, a mosaic of DNA sequences derived from several individuals, was first created in 2001 and has been used since then as the basis for comparison in genomic research. In 2010, researchers at the University of Washington, led by Evan Eichler, Ph.D., have discovered 2,363 DNA sequences that were not charted in the original human reference sequence. Since each segment of the human reference sequence is from a single person, it is possible that they did not possess the newly discovered sequences. This may not be surprising when you consider that about 80% of the original reference genome came from 8 people. The researchers found that some of these newly discovered sequences were common or rare in different populations and that some of them were present in different mammal species: chimpanzee, Bornean orangutan, rhesus monkey, house mouse, Norway rat, dog, and horse. The 1000 Genomes Project, an international effort to fully sequence the genomes of 1,000 anonymous individuals, and other genome studies are accumulating a considerable amount of data on the human DNA sequence that will be mapped to the original, and incomplete, reference genome. The process of DNA sequencing is largely technical and represents a significant opportunity for those who will be contributing to the latest reference human genome, which will provide valuable information for DNA technology. Questions and activities.
And that concludes the entire Chapter 8 on DNA fingerprinting.